In the previous video, I had discussed about the molecular composition of cell. If you haven't watched yet, you can click the I button to watch that video. Otherwise, the link is also available in the description box. Today, I will discuss about the cell membrane. Let's look at the cell. The edge part is the membrane. It separates inside the cell from the outside. If we zoom in, it looks something like this. Membrane has three important components. First important component is lipid, which is basically the structural unit of the membrane. Second one is protein, which is basically the functional unit of the membrane. And the third one is carbohydrate. Carbohydrate gets attached to various proteins and lipids and they modify their functions. Let's start with lipids. If we zoom in the membrane more, we find this kind of structure. It is called phospholipid. Each phospholipid has one head and two tails. The head is phosphate-containing group and the tails are fatty acid chains. The tails are connected to the head through a backbone. Phospholipids are characterized as either sphingolipids or glycerophospholipids, each structurally different. Glycerophospholipids have a glycerol backbone and sphingolipids have a sphingosine backbone. Phospholipid is amphipathic or in other words amphiphilic. What does it mean? It means it has a hydrophobic or nonpolar end and also a hydrophilic or polar end. The head groups are hydrophilic or polar. That means they have unequal distribution of electric charge. That's why they just love water and interact with water molecules. The tails are hydrophobic or nonpolar. That means they have equal distribution of electric charge. They just hate water and don't interact with the water molecules. The environment of inside the cell as well as the outside of the cell is aqueous. Water dissolves other substances. Then how do the hydrophobic tails survive? By creating phospholipid bilayer, where the fatty acid chains are buried in the interior of the cell membrane and the heads are exposed to the water. Phospholipid bilayer is made up of two phospholipid monolayer. Inside the cell, the fluid is called cytosol, which monolayer faces towards the cytosol called cytosolic membrane and which monolayer faces towards outside the cell called non-cytosolic membrane. The tails of this monolayer face each other. We know now that each phospholipid contains two tails. One of them has one or more double bond. That means these fatty acids are unsaturated. The double bond creates a kink there in the tail, making it harder for the chains to pack tightly. That's why membrane is quite fluid, because the kinks increase the space between the phospholipids, making the molecules harder to freeze at lower temperatures. Another one of them doesn't contain any double bond. It means these fatty acids are saturated. As a result, the chains are straight and easy to pack tightly. That's why the membrane is slightly rigid. So we can say that the membrane is fluid but slightly rigid. Now let's talk about the functional unit of membrane, the proteins. Based on position and function, there are three major classes of proteins: integral membrane protein or intrinsic protein, peripheral membrane protein or extrinsic protein, and lipid anchored proteins. The integral membrane proteins or intrinsic proteins are integrated or embedded into the cell membrane. They get permanently attached to the membrane. There are different types of intrinsic proteins. One is integral monotonic protein. It embeds into the cell membrane from one side, as you can see in this picture. Another one is transmembrane protein. Transmembrane means beyond membrane. This protein expands beyond the membrane. Transmembrane proteins are embedded into the cell membrane with portions exposed to the outside on both sides of the membrane. So we can say that transmembrane proteins span the membrane and extend to the outside on both sides. Now there are two types of transmembrane proteins. One is integral biotopic protein, which has one membrane spanning region, And the other one is integral polytopic proteins, which have many membrane spanning regions. Let's understand this again. The integral membrane proteins or intrinsic proteins have different types. One is integral monotopic proteins, which embed into the cell membrane from either side. The others are integral biotopic proteins or polytopic proteins. They both are transmembrane proteins because they span the membrane and expand beyond the membrane. The difference between them is the number of membrane spanning regions. Biotopic proteins have one membrane spanning region and polytopic proteins have multiple membrane spanning regions. The integral membrane proteins are amphipathic. Its hydrophobic portions are the neighbors of the hydrophobic fatty acid tails. Its hydrophilic portions are exposed to the outside. We discussed about integral membrane proteins which get permanently attached to the membrane, whereas the extrinsic proteins or peripheral membrane proteins get temporarily attached to the surface of the cell membrane. So they are not embedded into the membrane and they are not permanent. When they are needed, they come, associates with the cell membrane, directly being attached to the surface of the membrane or indirectly by interacting with the integral membrane proteins. They perform their function, it's done, they leave. I hope I made you clear about the difference between the intrinsic proteins and the extrinsic proteins. Now let's discuss about the lipid anchored proteins. They are located on the surface of the cell membrane, attached to the lipids embedded in the cell membrane. You may get confused between peripheral membrane proteins and lipid anchored proteins. The difference is that the peripheral membrane proteins get attached to the surface of the membrane and the lipid anchored proteins get attached to the lipid embedded into the membrane. So if we put the whole scenario of proteins of the cell membrane in a single picture, it something looks like this. So we know now that along with lipids and proteins, there are also carbohydrates. Carbohydrates get attached to the lipids of membrane bilayer and also with proteins of the membrane and modify the functions of lipids and proteins if needed. These glycolipids and glycoproteins together form glycocalyx, aka pericellular matrix, because it's found on the surface level outside the cell membrane. It basically acts as a cell coat. Cell membrane separates the inside of the cell from the outside of the cell. This acts as gatekeeper and barrier. Cell membrane is semi-permeable in nature. That means the membrane allows some molecules to pass across the membrane bilayer but not the others. Now let's understand which molecules can pass across the membrane and which molecules cannot. Small uncharged nonpolar molecules such as oxygen and carbon dioxide easily pass across the membrane because they are small in size and also nonpolar in nature. Small uncharged polar molecules such as water can pass across the membrane but slowly because they are polar. We know that interior portion of the membrane, which is basically made up of the hydrophobic nonpolar fatty acid tails, they don't like waters. Water molecules are small in size, so they can pass across the membrane, but because of the polar nature, it happens slowly. Large uncharged nonpolar molecules, such as the benzene, can pass across the membrane, but it also happens slowly. Because they are nonpolar in nature, so they can pass across the membrane, but because of its large size, it happens slowly. Large uncharged polar molecules, such as glucose, cannot pass across the membrane because they are large in size and also polar in nature. Charged molecules such as inorganic ions cannot pass across the membrane. Now the question is, if the cell membrane does not allow the transport of this kind of molecules, then how does the transport of these molecules happen? Here, proteins of the membrane play their role. Transmembrane proteins act as membrane transport proteins. Transmembrane proteins have membrane spanning regions through which these kind of molecules can pass through. There are two major classes of membrane transport proteins: channel proteins and carrier proteins. So some transmembrane proteins act as channel proteins and others as carrier proteins. The channel proteins form open pores through which the charged molecules travel in or out the cell. Ion channels, for example, allow the passage of inorganic ions from one side of the membrane to another side. The pores formed by these channel proteins are not permanently open; rather, they can be selectively opened or closed for specific molecules. The carrier proteins, aka transporters, allow the passage of large uncharged polar molecules. Rather than forming open channels, carrier proteins selectively bind molecules and then undergo conformational changes that open channels through which the molecule to be transported can pass across the membrane and be released on the other side. Now the question is, how do the channel proteins or carrier proteins become selective to molecules? It occurs by cell signaling. Cell signal is a process by which a cell interacts with itself, other cells, and the environment. Cell receives and responds to signals by receptors. Integral monotopic proteins, peripheral membrane proteins, and lipid-anchored proteins play a huge role in cell signaling. They send signals about the substances which are about to pass through the cell membrane. And the cell membranes and the transmembrane proteins become selective to molecules in response to the signal. This is how the cell membrane maintains the environment of inside the cell and the outside the cell differently, as needed to protect the cell or prevent cell death. I hope you found this video helpful. If you still have any query or doubt, you can put that in the comment section.